Okay, welcome back to this Genesis live stream and our next panel, Converging Science and Technology. So every year, Genesis mixes discussions about market and business trends, but also importantly, the exciting development in science, technology, and where they converge. You may be aware that this year is not just Genesis' 20th anniversary, but it's also the 30th anniversary of the launch of the Human Genome Project, and the upcoming session is a tribute to this game-changing initiative, reviewing the progresses it allowed in our understanding of the genomic base of diseases and the subsequent omics research developments. But equally importantly, this panel will also discuss the challenges it raised in our ability to deliver the new era of precision medicine. I am therefore delighted to invite our chair tonight, John Sterling, the editor-in-chief of Genetic Engineering and Biotech News, to lead this very important discussion in the next hour. John, over to you.
Elaine, thank you very much. As Elaine said, it's the 30th anniversary of the initiation of the Human Genome Project. Jen has been covering it this year. We're going to cover it again next year. And this all sort of leads down to the area of precision medicine, which is what we're going to be leading to today. The panel is extremely distinguished. I'm delighted to have them. So let me introduce them very quickly. So Sean Mackay is the co-founder and CEO of Isoplexis, which focuses on providing widely accessible cellular proteomics to speed personalized and curative medicines. Dr. Joanne Hackett is general partner at Izzy Capital. The company is a global early stage VC firm and advisory practice with an emphasis on the healthcare technology arena. And we're going to be talking a lot about technology today. Dr. Stephen Minger is the founder and director of SLM Blue Skies Innovations. The company provides expert analysis on next generation healthcare technologies, especially in the omics space and regenerative medicine. Dr. Mohammed Afshar is CEO of Ariana Pharma. I know we've covered them in Gen, which provides advanced clinical and biomarker data analysis to help companies reduce clinical and drug development risk. And Sumit Jamuar is chairman and CEO of Global Gene Corporation. His company's working on something very interesting. Years ago, I studied anthropology, and we're going to talk about one of the potential drawbacks of the Human Genome Project, and Sumit is involved in this. The company works with organizations around the world to collect and analyze diverse genomic and medical data sets from beyond people only with white European ancestry. Global Gene offers insights and technology platforms based on the global genetic data set to healthcare providers, biotech, and pharmaceutical companies. So we got a group of panelists from a diverse background. Let's get going. I've got a lot of questions, and later on, we'll take some from the audience. So my first question is very general. It's after the launch of the Human Genome Project in 1990 and its final draft in 2003. Tell me from each of your individual perspectives, what immediate impact did the completion of the Human Genome Project have on biological science and technology? Whoever wants to take that. Maybe you, Stephen? I think you need to be off mute.
Sorry, there we go. Sorry about that. You know, as a cell biologist, I mean, I was quite enamored with the idea that this technology was moving forward. But, you know, to be perfectly honest and maybe a little bit critical, at the end of the day, you know, $3 billion in one genome, and that genome was maybe, you know, 90%-ish of a genome. You know, there was a lot of hype around this that really didn't translate at that time into anything really useful. The one good thing I thought that came out of it was the fact that many of the academic groups, in particular John Sulston's group in Cambridge, adamantly opposed the idea that the human genome sequence should be commercialized, and stood up very strongly against Ventnor, against this. And, you know, John ultimately won the Nobel Prize, and I think that was very, very important for making sure that this technology would be available widely without onerous commercial licenses, which we've certainly seen in stem cell technology, and particularly in the realm of embryonic stem cell technology and cell-based therapies. But what it's led to, I think, is really very, very important. And I know we'll get into this later, John, the whole kind of omics revolution. You have to start somewhere, and it's easy to be critical looking back 20 years. We should have, not just done Ventnor's DNA. We should have probably, Sumit will address this as well. I think we should have looked at maybe a number of different genomes and not just one guy's genome. What an ego trip. But it's, I think it's an important milestone. And I think what it's led to now is what's really important. And, the fact that we're now not just genomics, but all the omics, which we'll talk about. My 2 cents.
Okay. Anybody else have a nickel to put into the discussion?
So John, I think, look, I just adding on to what Stephen said, it's incredibly exciting. And especially as we sit here having this virtual conference, thanks to the pandemic, when we talked about genomics at the start of this year, the scientific community, it was mainstream there, and people were going and saying, yep, that's great. And they would ask you the questions. What's happened with the pandemic and the development and the benefit that we've seen with, let's say, the vaccine development and other aspects, genomics has come into popular imagination. So people right now on the street, when they would meet you and they know that you're working genomics, they will ask you all sorts of really insightful questions, which, a few months ago they wouldn't. So what's exciting is the possibility that we can digitize ourselves starting with genomics, but other omics that come into play, build an identity about who we are, and then use that information. And that's where the promise is to be able to develop insights into who we are and how we have a productive life. And we benefit from things around, questions that we will ask is, can we make illness optional, right? And those are things where the promise comes into play. And how do we lead a productive life and a long life and a healthy life with people we love? And I think that's the digitization aspect and the insights that's coming in from the Human Genome Project. That's what the promise is. And I think what has happened recently is fundamentally transforming and accelerating that process. So I would not be surprised that when we look at healthcare in 5 or 10 years' time, it will be completely different from the traditional model that we have followed in the past. And I'll just pause there and let other panelists come in with their reactions.
One thing, Sumit, I'll add, I think it's exciting, both you guys, when you think about the impact on, John, on the industry, which you cover a lot over at Gen, there's I read a thing before that said, hey, people were trying to understand how quantum mechanics, mattered in life. And then they said, oh, a bunch of physicists talked about quantum mechanics. And they said, well, every computing innovation that happened in the last 30 years was because of quantum mechanics. So, okay, probably it has some meaning. I think there's a lot of meaning from the basic research into the genome that we're now starting to see. If you think about where things were in our industry 12 years ago, I don't think anyone really believed, other than the real clinicians and the scientists that understood the basis of it. I think the industry folks didn't necessarily believe that immune therapies were going to be, the number one cancer drug, and didn't believe that, gene editing and, changing people who had genetic variant problems was going to be a therapeutic modality today. It just seems so far off. Mm-hmm. And we're seeing that now be a reality. And that's all this recent therapeutic success, while I think it's scratching the surface, is based on a lot of those genomic insights. But what it's done is now there's so much investment in future therapeutics and very far-reaching things that we would never imagine. Well, we're working on some proteomic aspects and things. So I do think, like Sumit said, it's sort of, this is building on itself. People in not only the popular imagination, but sort of the larger, let's just call it investment community, governmental communities. There's a willingness to sort of build on this self-fulfilling prophecy that might not have happened if there wasn't this sort of impetus through the Human Genome Project. Mm-hmm.
Yeah, I'll just build on that by saying that I think one of the things for me that's been the most refreshing is, as Stephen said, what was it valued at as soon as it was completed? Probably not very much, as harsh as that sounds. Really making it mainstream. There's so many people that now think about doing a a commercial kit at home for Christmas sort of thing. It's become a little bit more mainstream. And while there are aspects that we can debate whether or not that's really genomics or whatnot, it's actually socialized it in a way that is actually quite exciting. And then what that's done is actually allowed a whole group of individuals who are interested in spinning out companies that have particular algorithms. There's a lot of tools and technologies now that are really, really impacting the industry as a whole, and this creating something that wasn't even here 10 years ago. So it's not only helped popularize and socialize what genomics really is, it's actually helped a lot of innovation and creativity come through and create jobs, and have a lot of return on investment, if you want to call it that. The other thing that I think is quite important, and this was just touched upon as well, is we're starting to realize clearly from the research and development that's been done with all of this data, is that there's really no such thing as one particular disease or therapeutic domain. And so obviously understanding more and more about rare diseases and starting to really get a grasp on the understanding that there's really no such thing as a common disease. And you're getting more precise now than we ever have been with targeting those treatments. And I think the whole premise and the whole belief that at least I have is that genomics is actually paving the way for us to think about ensuring individuals don't get sick in the first place. So currently we're treating sick people. How do we prevent them from getting sick? And then how do we actually move to a whole ethos of wellness?
Okay, very good. So based on the responses, I'm going to jump ahead a little bit. So we're all looking at precision medicine, and we're going to talk about its pluses and minuses in a minute. What are some potential benefits of precision medicine? Why are we even pursuing this? Sean?
I think benefits of precision medicine, like I think that sometimes the diagnostics, for good reason lags the therapeutic side, right? You make a therapeutic innovation, you don't know 100% if it's going to work. A lot of it becomes empirical. You might not know the mechanism before you start to see it to work. I mean, this happened in, a lot of the immunotherapies, right? Not necessarily cell therapies where you kind of distinctly understand the mechanism, but the cancer immunology drugs overall. And so why is precision medicine important? Because everyone thinks, wow, these are like miracle drugs, and they're ending things like metastatic melanoma. Well, the issue is even the most prominent drugs in the checkpoint inhibition space are at like 25% efficacy in most solid tumors, right? If that. And what we've done as a company, and also we're a single cell proteomics company, but there's also genomics companies doing the same thing in other avenues. We've focused on that sort of 25% of people that do well on the drug, and there might be other drugs available for those 75% people, which is the right drug at the right time. And like recently, we came out with data with a trial with Bristol Myers Squibb and Nektar showing that you could actually use our T cell assays in the blood and predict response of people who had durable response to checkpoint inhibitors. So the promise there is you had this drug that was a miracle drug. If you can partner a nice analytical assay, whether it's a genomic or a transcriptomic or a proteomic assay that tells you what patients will do better in the long run, you can just target those patients, and have a very high degree of efficacy. That's the promise. It has to be further validated in, hundreds of patient studies. And then those other patients who that drug is not going to work well with via whatever, their immune system or their tumor mutational burden or something else that's going on with them, can perhaps get other drugs earlier. And that's like a precision medicine in an ideal state is this sort of amazing triaging mechanism, right? If you could just have the right milieu of assays where you could figure out if patient already has the disease and they can get sort of translated to the right therapy, that would be amazing for society, because there's it's an obvious thing, but there's so many studies that say the earlier you catch and treat diseases for a specific sort of disease that they have, the better. So, and there's a lot more that other people on the panel on the genomic side could add to this, because there's been so many advancements in sort of genomic assays for targeted precision medicines that have totally changed the way we treat cancers.
I think you're absolutely right. And I think it's almost like it's allowing us to make a transition from, a binary readout of clinical trials into really trying to understand if you have, even in a clinical trial, if you have 3 patients that respond, is trying to understand why they respond. And now there are now a growing number of examples where with the right genomic characterization, one is able to try to understand the hypothesis which is behind the mechanism of action of a particular drug, and from what overall would have been just yet another failed clinical trial, actually understand what is working and transform it into a success. I mean, the ALK stories is a famous one, and there are others. And I think the beauty of this is that, for me, you were saying, the first milestone in human genome analysis was, once, transitioning also from the genetics into genomics. I mean, when I was a medical student, we had a lot of, work on the genetics and genetic diseases, or things like diabetes, etc. You were looking at particular genes, but suddenly this opens it up to every therapeutic area. And going back to what Joanne was saying, is then you look at it globally. And, now we can see, again, cancer is obvious, but, we've been involved in projects on Alzheimer's disease, where, novel genomic biomarkers can be identified from whole exome sequencing. Type 1 diabetes vaccine, again, genomic analysis is identifying groups of patients that behave in a particular way, etc. So I think it's really opening it up globally across therapeutic areas and allowing to transition out of this binary mode of evaluation.
So if I can just build on that, Mohammed, I think from my perspective, the problem has been is that, we were sold that genomics was going to solve everything. And as somebody who's, sat and listened to 10 or 20 or 30 cancer guys get up and talk about their favorite gene of a particular cancer, and, we looked for new mutations, we screened 50 patients with breast cancer, et cetera, et cetera. What did we find? Oh, we found the same mutations that we've always found. We didn't find anything new. As we know, and Sean just pointed this out, you can very, not precisely, but fairly rigorously partition patients into certain silos, all with the same cancer, but with different phenotypic and genomic phenotypic assays. And nevertheless, even with patients who quote unquote have the same form of cancer, some respond, some don't. And if you look at them, you can't really tell the difference between them at a genomic level. But there's some nice, interesting studies now that show that if you give patients the same drug, the ones that respond have a completely different microbiome than the ones who don't respond. So genome, in some sense, maybe doesn't tell you anything. And where I think, from my perspective, what I think the new revolution will be is this omics revolution, where now you build giant databases, not only of your genome, because in essence, your genome is just a book of letters with no paragraphs, no starts, no stops, no chapters. It doesn't tell you anything. You want to know which genes are expressed in which tissues, at which stage, and how a tissue that's involved in some sort of disease state, Alzheimer's, cancer, whatever, differs from the rest of the genome, which doesn't have that disease state. And so then that's transcriptomics. Sean's already talked about proteomics. We've got metabolomics, microbiomics, et cetera, et cetera. I think we need to build up a much more sophisticated, more holistic approach to how we treat individuals. And this goes back to John's question about, personalized or precision, medicine and diagnostics. All, I don't know, even B-cell lymphoma, which we've been treating reasonably well with CAR Ts. Not all patients respond, despite the fact that you can grow their cells up. They're all targeting CD19, and yet some patients respond, some patients don't. Even though we can grow equally potent numbers of cells, we can show on the dish that they work. You put them into the patient does not respond. Or they go into remission for a while and then their tumors come back. And it's not just a genomic thing. And this is where I think, the advent of big data analytics, synthetic intelligence, by people like DeepMind and others, building these massive databases of all this omics information, which is layered on top of one another, will give us humongous insight, particularly if we can do this in an unbiased fashion where basically, the machines are looking for correlations on their own without us guiding them and telling them what to do. So that's, I've listened to a lot of genomics talks over the year, and they all tend to be the same. We looked for what we wanted, and we either found it or we didn't, and we ignored all the other stuff.
Can I pick up some? I'm sorry, go ahead.
Yeah, so I just wanted to add to Stephen, and I think we'll get into more discussion about, what is fascinating is, you're starting with a set, and then, as we discover more and more, proteomics, transcriptomics, all these wonderful things, it all adds up to the insight that we need to build up. And of course, machine learning, AI, the convergence around that is really important. But I just wanted to step back and say, look, we spend about $1 trillion every year on medicine, right? On drugs. That's what we spent. We know that for certain drug classes, at least when I look at the data from 3 or 4 years ago, in oncology, we knew that 3 out of 4 patients would not respond to it. What we have found with using these technologies is that we are able to personalize that experience, and that ends up being far more effective and specific to the individual concern. So if you go back to the summary around, John, what you started with, I think what excites me personally is the definition of precision medicine, which is right to the right person, the right drug at the right time in the right dosage, right? If we do that. And if you push that paradigm even further, given all the insights that we're building up, can we then change that and using that to move it from a paradigm where I fall sick today, then I have a treatment which is then diagnosed, to a paradigm where I say, look, these are the potential risks which are there. And again, there is still work to be done to get to that stage. And then I can stay healthy for longer because I can avoid disease altogether. And I think those are the themes which are really attractive. So from efficacy and efficiency to also moving to a different paradigm of healthcare, because the traditional model, unfortunately, if $1 trillion, if 40 to 45% doesn't work, we basically in number terms is 400 to 450 billion dollars is being spent on things which don't necessarily work. So, and what we are all interested in the room and in the industry is to say, look, how can we be more productive and more effective in doing that? And can the insight that we build with these technologies enable us to do that? I'll just pause there and
Yeah, so I would just add to that. Obviously, we talk about this pandemic, and obviously it's an awful thing. We're all experiencing, horrible things because of this, and the number of deaths associated with it are quite large. But how many people a year die from adverse drug reactions? We don't talk about that. That's something we have a little bit more control over rather than a global pandemic. And it's not even just the people's lives. There's obviously a huge amount of burden on society, as well as clearly cost as well. And, as previously discussed, no one is trying to actually, move slower or harm people, but we need to actually be able to understand what's happening inside to be able to make those inferences. And the other thing I do think that I think many people have echoed this, genomics is definitely only one data point. I do think the hype over genomics was probably overextended at the very beginning, that it was going to be the answer to everything. But again, I think that the thing that it's probably helped socialize is that there are these other omics that are equally important as well, and that comprehensive picture is something that we really should be aiming for. So while it's probably, again, popularized the concepts that have come after the fact, it certainly has been a forefront in that. And I do think that in this report earlier this year from the FDA, there's 50 known gene-drug interactions. Why are those not standard in every pharmacy around the world? Why is that not something that we're thinking of doing in the first stages? And then I'm sure we'll get into it as a discussion point later on, but where are the industries that are actually at the forefront of being able to change the way that we think about this? The insurance industry, why do we think of genomics and multi-omics approaches as a negative thing? Why is that actually not part of your healthcare screening package so that your employer can actually monitor and allow you to have a better quality of health as opposed to penalizing you for something that is lurking underneath? So I'm sure we'll talk about a lot of that stuff as the panel goes on, but I think the personalized and precision medicine approaches, we're getting closer to actually realizing them, because we're now finally starting to aggregate a mass of data that's allowing us to look differently, but also there's new tools and technologies that are allowing us to do that as well.
Okay. Is that fair to say that maybe the promise of precision medicine is actually to enable personalized medicine? So if you're outside of public health, I mean, when medicine is supposed to be personalized, and in a way, the genomics allows you to have the tools to really personalize and deliver on that promise. And indeed, if you have access to gene drug panels and etc., then actually that enables what you're initially supposed to do, as taking care of a particular patient in a specific way.
Mm-hmm. This business of big data analytics and artificial intelligence, we're seeing that in Gen everywhere, from drug discovery now to biomanufacturing. It's everywhere. And we're definitely going to come to that later. But Joanna, I want to come back to you for a second, because again, we're talking about 30 years ago, they started this project, and we're talking about omics technologies. And people usually refer to genomics, transcriptomics, proteomics, and metabolomics. Since you work for a VC firm, plus you're a scientist, you talk to people who know a lot about science and people who want to learn more. What do you tell them kinds of therapeutic insights have been gained or can be gained by using these methods in life science research and clinical applications?
Yeah, so I think the two biggest areas that we've probably touched upon already is obviously cancer and anything to do with a rare disease. And I think that, oncology is certainly a bit more complicated than rare disease for a variety of different reasons. But I think I've always said that if you're willing to amass any sort of data, you're definitely going to find insights. The biggest problem is there's two different groups of individuals that end up having this type of test. And most of them are actually sick. Very few of them are healthy. The healthy ones are not having the full comprehensive testing that obviously you would need to garner those insights. So we end up having a sick population, which allows us to only look at particular mutations. And then the healthy population that we have is there's very few of them. And therefore we don't actually have a reference to really be looking into. So there is a difference between that. And the other thing that is important, which I know we're going to be talking about later, is the ethnic diversity. So it's very difficult to be able to make a blanket statement about a particular drug or therapeutic and how that's going to impact X population if you don't actually have that evidence. So again, while I do think that it hasn't been that long since this has been, the Human Genome Project has been finished, the insights will definitely take a long time to actually to bear fruit. But that's not to say that we're not seeing things already. We're seeing things all the time, which is quite exciting. New technologies that are able to look at structural variants. That's actually extremely important for rare disease. Being able to build panels that are put into healthcare systems that are actually occurring on small portable devices. This is technology that we wouldn't have had we not actually been able to amass the population genetics that we currently have today. So I think, generally speaking, it still ends up being rare disease and cancer, mostly because that is you can see the phenotype and the genotype effectively. But I do think that there are many more targeted approaches. We see a lot more of industry coming together in consortium to actually look at particular populations, whether it's early onset Parkinson's or whether it's Alzheimer's. There's a lot of interest in trying to amass that data. The thing that very often is missing is the fact that you don't take everything at the beginning. And that generally is based on the fact that we don't know what to be asking for. So sometimes we say, let's take all information and all data because it'll be useful. It's actually not necessarily the case if it's not in the right format, if it's not structured, if it can't be federated. So the whole AI ML section with respect to big data is actually, people think it's easy to just grab a bunch of data and make insights. If it's not formatted and standardized, and if it's unstructured or structured, it's absolute mess to try to deal with. So we've actually seen, I think, a retraction with respect to deals for data acquisition, mostly based on the fact that industry has been burned. They've actually spent quite a lot of money trying to amass this data and realizing once they get it through the front door, it's actually not what they expected it to be. So I think people are still playing it a bit safe and looking into cancer and rare disease as opposed to actually trying to proactively find these other populations, mostly because they would have to be funded probably privately.
Mm-hmm.
I would absolutely second that. And I think that, on what Joanne was mentioning and Stephen earlier on about, the vision is to be able to pull all this data together. But then the challenge is to be able to analyze that on a global way. We're just looking earlier on this morning on a very simple clinical trial, 30 patients, and we have some genomic and RNA expression data at multiple time points. There's more than 7 terabytes of data. You have several 100,000 columns of data for each of the 30 patients. and developing the right tools to be able to analyze that in a and obviously you can't use statistics when you have such a you have these wide matrices rather than long matrices. So really developing the right AI to be able to handle these very wide matrices, absolutely critical.
So I would say you guys have provided me the perfect segue for the work that we are doing anyways, because I agree with you. I mean, look, if you think about it, there are three really important aspects to the things that you've discussed right now. The first and the most important is the ethical, paradigm in terms of making sure we have the right IRB approvals, the ethics consent to be able to do something with whatever we are curating. And that is the starting point. The second is what I would call the scientific point about saying, what are we solving for? Whether is it allelic architecture, whether it's understanding the disease, and how do we want to understand it? And then the third is actually creating and curating the either retrospectively or prospective data set. And the fundamental thing starts with, it's not just about the sample and the genomic or the RNA, et cetera, but it's not just the sample and the sequencing of that, but it's also the most important thing we find in all our engagements is the phenotyping and the depth of that phenotyping and standardization. And that is why, when we started, we deliberately went to Indian subcontinent for the simple reason that India has 4,500 different ethnicities, which is almost 50% of the world's ethnicity. When we look at discoveries that are happening, whether it's PCSK9 from the Icelandic population, whether we're looking at SCN9A from the Inuit Canadian population, as well as the South Asian population, you're looking at very specific cohorts where the phenotype guided the genomic studies. And building that in a form where one can standardize that and then put it on global, very clean ontologies, the norms is really important. And that's what, for example, my team specializes in. That's what we've built over the last few years. So I agree with you, because sometimes cleaning up the data takes so much time that you cannot get things done. And that's what people are attracted by, is the ability to get at scale cohorts with deep phenotype, including labs and other aspects which are needed, electronic health records or health records where possible, because that's what adds the depth and the value of that. And so I would say that, look, I'm glad people recognize how tough it is, but also the fact, that's where we find that, having that ability to execute it is what will solve the problem, because we have to solve the problem to make it useful. And some of the points that Stephen mentioned around not being useful is because we sometimes do not have the reference on what we are comparing against, which then leads into some of the paired conversation, which I'm sure we'll get into. But I'll just pause there. But thank you for providing a perfect segue for what we are doing at Global Gene Corp with our esteemed industry partners.
Almost as though we set it up for you.
As always, good partners.
We aim to please.
Thank you, John.
So I thought what's really interesting, again, Jen, we really focus on tools and technologies, and we were just talking about all these different omics techniques. And I know we have a number of scientists listening here, but for others who may be coming new to this field, what types of tools and instrument systems have become mainstays of omics research, and what kinds of information do they provide that are leading to some of the therapeutic insights that you people have found in your work? Mohammed?
Yeah, I think that I think really over, if I look at over the last 15 years where we've been working in this field, I think the technologies around the rapid sequencing have I think they are really impacting the way, again, these techniques are getting into both the drug development, but also on the therapeutic decision support. So it's even doing a whole exome sequencing in a it's a few $100s, and it means that it can be integrated if you can analyze the data. That's a big if. It can be very easily integrated into any clinical trial. And in terms of the looking at the therapeutic decision support, I think all the array technologies that allow you to do these very rapid panels, I think they have had a huge impact, because again, the real cost is a few dollars, to actually do the panel. So I think that's where the technology is becoming more and more efficient, and that is then it enables it to go to cross the, go not only be a sophisticated research tool, but really now get into the mainstream.
Sean, you definitely in the technology space.
Yeah, no, I think I think the sequencing, I think cost and rapidity of sequencing is a huge part of it, right? And throughput. If you look at, and again, I'll focus on sort of cancer. It's not the only place we focus, but it's a big part of it. Two aspects of things that have been accelerated by sequencing is the bulk sequencing and the ability to sort of understand, the tumor profiles in a rapid fashion that can be applied clinically, based on a cost and sort of data analysis perspective. There's still gaps, I think, that can be solved, as sort of Stephen mentioned, by other omics things, which is where we play in, but that is helpful. And then therapeutic development, of course, with, if you're thinking through people that make new cell therapies and things, which we also help with. We do a lot of the sort of quality checks in process sciences for these sort of advanced cell therapies. You wouldn't be able to do any of this stuff without the advancement of genomic sequencing and synthesis and the like. If you think of the other modalities that matter, I do think, we're now seeing not just sort of baseline tumor pathways as mattering, which that is particularly relevant when you're measuring various genes like, tumor suppressor genes or oncogenes that can be targeted more easily with small molecule drugs. But as the drugs evolve for precision medicine, like immune therapies, I think the challenge and the opening sort of the gates for other technologies has been that, these other cell types, let's say like immune cells, are a bit more plastic and dynamic in nature. And what happens is, you need other answers that couple with, let's say, what the tumor looks like from the genome level, that answers perhaps what's happening dynamically, like from the translational or post-translational level at the proteome. And that's where we're coming in. There's other technologies. What we do is, we look at things like the secretome and the intracellular proteome of each single cell. And there's other technologies like, single cell sequencing and other, optoelectronic single cell sorting. Each of them are playing a part in analyzing, what these more, I would call them dynamic or proteomic, plastic nature of immune cells are doing. And I guess when I just couple that with why it matters on top of the genomic layer is, what we've seen, just paper coming out in Cell 2 weeks ago using our platform, you could look at a large amount of COVID patients and see that small subsets of cells that were dysfunctional proteomically, like these monocyte or innate cells, were predictors of which patients would go into the ICU with COVID very early on. And that's not necessarily going to be made a diagnostic tomorrow, right? Because that's more mechanistic, and there's cost considerations and all these other things. Are there enough of an installed base to put this into a lab? But the point is you can imagine in 3 to 5 years that other assays other than a straight bulk genomic sequence could be additive to the equation of sort of what's happening dynamically in the patient, similar to what was mentioned on the panel before. And then, how does that relate to best therapy to give? Or if you can predict things that are dynamically happening via proteomics earlier, perhaps you can treat when the disease is also very early and they don't have to, let's say, go into the ICU with COVID, or when they have an earlier stage cancer that you can treat with immunotherapy. So I would say the bulk to these single cell tools, which you guys profile a lot in Gen, that's evolved, because I would close with the interesting thing we found in these cancers is we talk about heterogeneity amongst patients, but tumor cells and cells of the CNS and immune cells by their nature are plastic and heterogeneous. So, capturing some of that sort of plasticity and that heterogeneity at the proteome level has evolved into maybe not a diagnostic need immediately, but a seeming diagnostic need multiple years out. So I'll stop there.
Hey, John, if I could follow on from that with a couple of examples. So I totally agree with Mohammed and Sean. I think, high throughput sequencing at a really reduced cost, but really rapid sequencing has fundamentally changed a couple of fields. And the two that I would point out, one is in the T cell space, in the CAR T space, let's say, immunotherapy space using T cells. The big problem we face in addressing things outside of CD19 on B cells is looking for tumor antigens that are specific to the tumor, which are unique from normal cells. And that's going to be very difficult to do. And particularly going to solid tumors. So, whether we go to tri or quad specific CARs that all have to be engaged before you see activation of T cells is, that's a big reach. But T cells themselves have T cell receptors, and they're used to seeing tumor antigens, which can be intracellular, which CARs cannot deal with. So I think high throughput sequencing of peptides that T cell receptors, TCR, gives us a whole different advantage in tumor targeting that's over and above kind of, genetically engineering a target receptor, that we artificially think is on a tumor cell that's not on. So, but this relies on a lot of sequencing. I think a more important aspect, something I follow quite closely, although I'm not a microbiologist by any means, I'm a neurobiologist and stem cell guy, is, our understanding of the microbiome, particularly in the gut, has completely changed in the last 5 years, because prior to the advent of high throughput sequencing at a really, really low cost, we didn't really know anything except for a few things that we could culture, and 99% of the stuff that grows inside us sits in an anaerobic environment. And even if you can get live bugs out, by the time you put them into culture, they don't survive. So really, the idea that we're actually being able to understand population dynamics within different regions of the gut across thousands of species. I mean, we still don't even know how many of these things live inside us, on us, in between us. Sequencing has given us insight into this where other conventional techniques don't work. And again, you're actually sequencing unknown populations where we have to build up phylogenetic trees based on pre-existing genomes, and then build from there. But this is still early days. But I think again, it's going back again to the idea of precision medicine and diagnostics. How much of our, just focusing on the microbiome in our guts at the moment, how much of that predisposes us to health, and how much of a change in that symbiosis, communal symbiosis, leads us to Parkinson's disease, or to Alzheimer's disease, or to type 2 diabetes, or to inflammation. We just don't know. And without the advent of sequencing, I would argue we'd still be completely in the dark on this. And I know there's a lot of skepticism within the scientific and investment community about microbiomics, but these things didn't evolve inside us for nothing. And I think that the data that's now starting to come out is really quite dramatic in terms of its influence on disease and health, longevity, et cetera. So I'll stop there. Thanks.
By the way, just as an aside, we did an article a couple of months ago. They studied the microbiome on the Parisian subways and the New York City subways. And lo and behold, they were basically the same. I don't know how far that's got, but that just amazed me. I've been to Paris many times. I love the city. Water's different, food's different, everything, but the microbiomes seem to be the same. So there's a PhD thesis. So
John, that's thanks to Mohammed, who keeps traveling between Paris and
that's the thing, yeah. But John, I just wanted to add to what Stephen was mentioning. I think that's where we worked on a project looking at the response to a particular drug, and looking at the genomic, but also the gut microbiota. And I think that you get really interesting hypotheses which are linked to the predominance of particular species, etc., and the way they actually then are linked to the outcome. And this is beyond absorption or whatever. And you can there are similar stories in oncology. But again, I think then that's where having the right AI technology becomes absolutely crucial. Stephen was mentioning that we keep looking for something. Either we find it and we're happy, we don't find it, we ignore it. But I think in this particular field, if we, it has become really critical to develop these explainable AI tools. And we've been working on this, John, as you know, for more than 15 years now, and really trying to look at these unsupervised machine learning techniques that can identify, again, hypotheses. And hypothesis is not something negative. It means that you can then build up on it from this very wide data. And I think these are approaches that will have a big impact beyond what has been seen in deep learning, etc., where when you relatively understand relatively well a particular concept, etc., then you can just launch your deep learning, machine learning, and it finds a model. It doesn't explain why, but it works. I think the challenge in the approaches like the understanding how gut microbiota works is that you need the right explainable AI that is able to have a dialogue with you to say, look, these are the features I found. This is why, and what do you think? And maintain consistency in the reasoning, and then just move up along the scientific discovery, helped by a tool that manages these very wide matrices.
Yeah. Now it's interesting to talk about the microbiome. Whenever we do stories on Gen and we put them up on the web, they usually come within the top 5 of the stories we do. So that definitely is a hot area. So I want to jump ahead a little bit, and I'm picking up on what Stephen said. Having covering biotech for 36 years, Leroy Hood, I'm not doing it now, I guess I am. Our January issue was the 40th anniversary. And I asked Leroy Hood to do the key piece, and you guys should really read that. What he went through to get this academic stuff into the industrial world, it's quite a story and things like this. But there've been, I've seen great scientific advances, but as Stephen said, I've seen hype galore, at conferences and every place and things like this. And it's just my feeling, and maybe just me, that with the way people talk about precision medicine, it should be further along than what we've heard and about all these great advancements. So what do you folks feel are the major roadblocks and barriers that are holding precision medicine back from having greater acceptance by the medical community and not being applied more broadly in clinical applications to treat disease? Anybody?
I'll kick this one off. So I've often said that it's not, we probably have most of the tools and techniques to actually do this. And I do think that there are actually quite a lot of advancements that we're not even aware of, simply based on the fact that there's no investment in the plumbing. The non-sexy stuff, it's the data connectivity, it's the structuring of data. That stuff is just absolutely not sexy, and people don't invest in it. You could write to the MRC and ask for £5 million to do some really interesting research, and you'd probably get it. If you ask £5 million to actually build a data infrastructure within a particular university or a hospital, not going to happen. It's the non-sexy stuff that we've not invested in. And I think there's lots of innovations that are actually probably sitting there ripe for the picking. We just actually haven't done that connectivity piece. And some of the discussions previously when you were saying sequencing and the sequencer and a lot of the stuff around those platforms is really what has pushed this forward. Think about imaging as well. All of those imaging facilities around the world that have spent hours and hours and hours. If you can train an algorithm, and we've seen this, obviously, it works really, really well. But imagine connecting that to a scan, to primary care data, to the number of steps that you take to your Tesco card, all of that stuff. It's that connectivity that we just simply have not invested in. And unfortunately, we've actually seen the repercussions of that based on this pandemic. We're seeing this every single day, unfortunately. But what that has also highlighted is that there are solutions for this, and we just actually have to get a little bit more robust about investing in the plumbing.
And if I may just add to, the plumbing, and Joanne has fantastic experiences as a leader of one of the iconic programs that we all talk about in her previous life. One of the things which drove us to do what we do is partly frustration, which Stephen expressed, which, my co-founders and co-chief scientific officers, so Dr. Jonathan Picker, he's a faculty at Harvard. And one of the reasons he got was he was frustrated by the pace things were happening. And this was about 8 years ago. He said, human genomes happened. He's a clinician. And he says, look, we had this promise of what's going to be delivered, and things are moving too slow. Because when you think about genomics, the first phase about from $2.7 billion, how do we reduce sequencing to a few $100s and lower? So that was the first phase. The second phase phase was what are the tools and technologies and analytics platform do we need to enable us to analyze that data. The third thing was about, use cases. And then this is now the focus is on data, which is the plumbing and everything else, which is there. I'm still staggered that 80% of all data, when we look at the recent study in the Nomad data set, 80% is from people of European ancestry, right? And here is the technology where we are saying everything's going to be precise exactly to Stephen, who you are, to Joanne, and to Sumit. And yet we are saying we don't necessarily know who you are. So I'm going to compare Sumit to Stephen and hope that I get the answer right? And that is one problem which leads to a lot of other challenges in terms of, if there's no so what to what I do, it doesn't make a life-changing difference and it's expensive, then I'm going to say, well, let me postpone that. Or the payer will say, why should I pay for that, right? So all these things come into play. So one of the biggest roadblocks we've seen is not insufficient understanding with the right level of detail, whether it's phenotype or EHRs in the standardized format, with the right genomics and proteomics and microbiome and other aspects for either populations or for disease, right? So that's something which is really, if I had one point to pick, that's the starting point. And the second thing I would say is about talent and training. When you go to a clinician and you say to them, hey, there's a wonderful thing called genomics, which is going to solve your problem. But by the way, it's going to complicate your life because things keep changing every day, right? You have a great doctor. They do not have time. They want to serve their patients. They do not have time to learn something new, which is coming. So it's how do we deliver that from a digital, in a platform which is easy to use, which makes their life easy for them to adopt. And that feeds into the training when we set the next generation of doctors and healthcare professionals coming in. How do we imbibe that and coming into play? So it's, this technology side, and then there's also, how we think about the end user experience. And the ultimate thing is about the power of genomics is about giving the patient or giving the individual the power over their healthcare. And it's about how do we enable that to happen and put the tools and techniques and other things. And that's what this technology promises. That's what I'm excited by personally. So I just wanted to highlight, these are some of the thoughts that we have around what's stopping it from going there. Good thing is post-pandemic, things are accelerating. People are focusing on the right things and people are building on that. And we are all beneficiaries of it.
So I mean, just to add just to one point about what you were saying about the frustration about the training for the clinicians, etc. What's funny is that you have the same thing for people that are developing drugs. I mean, if I take oncology, which is where the space where genomic has had the largest impact, you still have clinical trials that are launched where there are only just a few markers that are tested, and when you ask why. And people are saying, well, what am I going to do with all of this thing? How am I going to analyze it? How am I going? It's just going to make my life more complicated. So I think education is going to be important, and getting the glue that Joanne was saying, the infrastructure that says okay, I'm going to pull all of that together. I am going to also provide you the tools to automatically analyze this and help you so that it lowers the barrier of actually adopting it. It's going to be absolutely key, I think.
So in looking at a lot of articles about this, two major things that kept coming up questionable were one, reimbursement, and Sumit, you mentioned that, and also physician acceptance. How big a role, are we far enough along where physicians understand this technology, or is this part of the training that they need to do to incorporate it into their practice, number one, and then say, hey, am I going to get paid for this? Are those still, is that correct? Those are two major issues?
Yeah, I mean, if I can address that, because GE, as early as, I think, 2011, 12, when I was chief scientist at GE, we established we acquired a precision diagnostics company with the whole idea that we were going to do precision diagnostics around a particular form of cancer, non-Hodgkin's. And, quite frankly, it just never took off. And the biggest reasons for that, from what I understand, reimbursement was a big one, because, they were like, well, our clinicians are good at diagnosing this, convince us that your platform really provides us with any additional information, and that, insurance companies will pay for it. And of course, at that time, they were very, very reluctant to do so. So that business ultimately kind of almost completely disappeared. On the training side, as somebody who's trained thousands of medics when I was at King's, I was very adamant, as were a number of my colleagues. I mean, King's is a big translational medicine. It's the largest medical school in Europe, and at that time, our dean, who was also a transplant immunologist, his ethos was, we have to do research-led teaching. Don't pull out Leninger and don't pull out all these old textbooks and stuff and teach these guys the same crap that they've learned for the last 50 years. Teach from your research. And so many of us had courses. I had a course on regenerative medicine with over 100 students, half of whom were medics. That was led by cardiologists, by transplant surgeons, people, treating patients with type 1 diabetes, a whole bunch of neurologists, et cetera, et cetera, et cetera. And, our real goal was to make these guys clinician scientists, and let our teaching inform their clinical decision-making. And I know that doesn't happen in a lot of medical schools. And, in talking with students that I trained years later, who are now consultants and stuff, they don't have time to keep up, particularly in the NHS, and I've talked to a couple of them after, COVID has happened, and they can barely keep their nose above water. And so the idea that these guys are constantly reading the literature and they're taking in all this new information, I think in many ways it's a little bit too much to ask. They just don't have the time. You have some clinicians that work in big medical schools where they do research, and maybe those guys are a little bit more able to do this. But I think your average clinician who has a busy practice it's just impossible for them to really keep up with this. And so they always go back to what they know. I mean, we're all conservative in our behavior in that regard. And so I think this is a real struggle to constantly be pushing the medical community to take on new things. I mean, immunotherapy for many, many years, based on T-cell therapy, people laugh this off, and the uptake has been slow. It's taken a long time for Novartis and others, Juno Kite, to get oncologists to think about this over and above traditional chemotherapy, radiation, and transplant. It takes a long time to change people's thinking. And I think that's a really big impediment to embracing these new technologies and make them part of standard clinical practice.
So we have about 2 minutes left in the session. Very quickly, with the pluses and the minuses for each of you, briefly. The promise of, is there a real future for precision medicine in healthcare? And how soon can we get that? Quickly, Joanne.
Yes, and we're there now.
Okay. Mohammed?
I would say absolutely yes. And I think that the frustration that people are struggling to access all this literature, etc., which is available to clinicians, and that they need to change their practice, is actually an opportunity for tools that can bring that together for them in an easy to digest way. So
I love that
yes.
positive spin. That's good. So.
Yeah, so I think as we are all deep into our areas, one of the things we have to step back and say, look, I'm very optimistic, and I believe that this is what's this is here, and it is the future. So when we look at the next generation, people are going, next generation will say, you guys were so medieval in terms of how you dealt with patients and with disease. I can't believe you cut stuff out, and I can't, you've got stem cell which could have sorted it, and I can't believe that you gave harmful chemicals just in the hope of something being sorted out. And I just want to end with a reminder of what just 20 years ago or just 25 years ago, when you look at internet, when it was coming in, we didn't even know things which would exist. We wouldn't even know that we would be having a Zoom call virtually to have this session, right? And that's the stage we are at, where things are happening. This is the late 90s, 99 of internet or whatever. And in the next couple of decades, things will arrive which we can't even imagine today. So I just end on that really optimistic note, and why we're excited.
Well, I'm a big fan of Frank Sinatra, and his big song is The Best Is Yet to Come, and I agree with that. I want to thank you all for an incredible panel. I look forward to talking to you for Gen articles. This was really great. Thank you for being here. Aline, turning back to you.
Thank you, John. And I know you didn't ask my advice, but I completely agree with this optimistic note, Sumit, you gave, and all of you. We're just at the beginning of it. And there's lots of, if I remember correctly, investing in non-sexy technology and also lots of education. And I think it's to bear in mind that next generation is coming in. And as you said, they will see things very differently and probably see this difference of paradigm that will make precision medicine happen, hopefully. So a huge thank you for you all. It was a really, really good discussion. I really enjoyed it. Thank you, John, for brilliantly chairing it. That was amazing. And so it's just to remind delegates that there is still another session in half an hour. I hope you join, which is our usual Genesis closing session, wrapping up and also looking to the future of what the industry will look like in 20 years' time. In the meantime, half an hour for more networking if you want to, and see you later.